Hi Hinton Scholars. This video is about our third Hinton lab, cellular respiration. I handed you a copy of this protocol in class today. If for some reason you didn't get one, pick one up tomorrow or today, depending on when you're watching this, but before you go to Hinton. I'm going to walk through it, but this is not a substitute for you reading it yourself and annotating it yourself. A lot of people didn't get to collect all their data at the last lab, and I think part of that was feeling a little bit of confusion about the steps that needed to be accomplished. And while this lab I feel like is a bit straightforward and you've worked with the equipment before, I still want us to walk in prepared so we can make sure we gather all of our data and get to the really fun part for this lab, which is the open inquiry part, because it deals with like crickets and mealworms and different type of beans and that sort of thing. So you want to be able to get to the open inquiry. So you'll read all of this intro. I'm going to point out that the first investigation we're doing will all do the same investigation, and that is trying to answer the question, do peas undergo cellular respiration before germination? So germination, what is that? You might not have heard that word before. Germination means when seeds start to grow. So we're going to be working with something called mung beans. They look like this. They're just little green beans. And if I had germinating mung beans, what I would see is that they had been soaked in water and they're starting to grow, like they're starting to sprout. They might be a little bit bigger, like you can see that they're starting to grow. So it says here that it is known that peas undergo cellular respiration during germination. So we know that when they're germinating, they're doing cellular respiration. We're trying to see when they're not germinated, are they doing cellular respiration? So like these sitting in my hand, are they doing cellular respiration or not? So we're going to gather some data to try to answer that question. Think in your head what we might measure to see if cellular respiration is happening. Giving you a minute to think in your head. CO2? Yes, CO2. We can measure CO2 levels to see if cellular respiration is happening. Is it going to go up or is it going to go down? CO2 is going up in cellular respiration, like we're exhaling. Cellular respiration is happening. We're exhaling CO2. And just like us, just like we exhale CO2, plants, animals, any organism that's living, if it's doing cellular respiration, it's going to be producing or releasing CO2. So we're going to use some equipment that is going to look familiar to you, and this picture here represents it pretty well, but I have the probes. This is what I was waiting for to make this video. We use this at the Saturday session. We use these Nalgene bottles, these plastic bottles, and a CO2 sensor to detect the amount of CO2 in the bottle. You worked with spinach, and in class soon we're going to do another spinach lab, but this time at Hinton, you're going to be working with some other things like the germinated and non-germinated mung beans, and some other fun things in the open inquiry. So you've worked with this probe before. You know that you can clear it by waving it in the air. Don't blow in it right before you're trying to do a recording because that kind of throws off its CO2 detection skills. It needs time to kind of like settle out and zero out. But you'll put your ungerminated mung beans in this bottle. You'll probably measure the rate of CO2 of them first. Then you'll put your germinated mung beans in, measure their rate of CO2 production, analyze it, decide, do ungerminated mung beans do cellular respiration? And then after you've done that first kind of set lab, you'll do your open inquiry lab. So we'll have options of things to choose from. Chances are not everybody's going to be able to do the crickets. Everybody's going to want to do the crickets. But do not fear. We're getting some crickets and some mealworms to bring back to the classroom. And you as Hinton Scholars are going to run this lab with the rest of the students next Monday and Tuesday. So we'll have the crickets and the mealworms to use then, even if you don't get to use them today, tomorrow, whatever day you're watching this, at Hinton. Okay? So even if you don't get to do crickets, there's other really cool things to pick from, like different types of beans, like different beans, like bigger beans do more cellular respiration than smaller beans. That's another thing that you can investigate. Okay, back to the protocol. All of the materials are listed here. And like I said, I want you to read all of this. Read and annotate, okay? So that you know what to anticipate. So you know timing, so you know what's going in where, how to measure what. You'll be using the lab quests again. I don't have one like they have at Hinton to show you how to use it. So if that was a limitation for you last time, make sure you're asking somebody there for help for how to get the lab quest going. We'll have a couple of mentors, a couple of OB students who did AP Bio last year and did Hinton that you can ask, and you can ask the lab assistants, and you can ask your tutors like George and Haley and Bing. They should be able to help you with the lab quests. So read, annotate. I want to see everybody bringing their own copy of this to Hinton. I'm still flipping through. You'll see that there's a place where we're going to record temperature. There's a place for you to record the rate of respiration. How am I going to get that rate? Rate would be change over time. Okay. Parts per million per seconds. I think that's what you're going to be measuring it, and that's what the units are listed here. Now that reminds me, as you're collecting your data, it's going to be collecting on your lab quest, and you'll save your data on your lab quest, but you also must manually record it. You'll receive a paper that looks like this. There are columns for you to record in. Run 1 will be your non-germinated mung beans. Run 2 would be your germinated mung beans. I think that's the order you'll do it in. So you're going to record your data here. In the protocol, it tells you how frequently to record the data. Like, is it every 5 seconds? Is it every 2 minutes? Is it every 5 minutes? Is it 425 minutes? So you have to pay attention to how frequently they want you to set your lab quest to collect the data. Okay, you'll also notice that there are questions here at the end. It is the expectation that you answer these questions before you start your open inquiry, just so you know. And in terms of the way that you'll be graded on this lab, it will be very similar to the last one. So I'll want to see in your lab notebook, don't forget to bring your lab notebook, that you've recorded your question, your hypothesis, your independent variable, like what you do for the open inquiry, as well as all of the data for the germinated versus non-germinated mung beans. Like, I should see this in your lab journal too. I'll be looking for the data that you collect, your data table. Okay, and I'm going to say again that you will be, hi, how are you? 1 second. That you will be, okay, that you will be running the lab in class on Monday and Tuesday, okay, with the other students. So just keep that in mind as you're going through this, so that you're paying attention to everything that's happening, so that you know how to help the students in class. We will have a slightly different lab quest that we're using, but the basics of the lab and the understanding of the lab will be the same. Let me know if you have questions. I'll see you soon.